Good afternoon to everybody, and welcome to this new debriefing. At France-Soir, we've been interviewing over 500 doctors, legal advisors, scientists around the world since the COVID era. We've interviewed people like Robert Kennedy, Jay Bhattacharya, Robert Malone, Ron Johnson, Americans that have influenced and that had a scientific discourse that has been cancelled. And the intention behind it is that we believe that the scientific discourse needs to have a forum where people can actually hear what these individuals have to say beyond what the media say about them. And today we're going to have on this show, and I'm really very grateful for him to make himself available, Professor Wafik El Deiry. He is a distinguished physician and scientist, medical oncologist from Brown University. He has several discoveries for drug discoveries, several scientific publications. I believe he has over 100,000 citations, 108,000 exactly. And he is joining on this show to talk about his work, his discovery, but also what he had to go through in terms of censorship, what he had to go through in terms of hassling. And so please welcome Professor El Deiry. Good afternoon, Professor, and thank you very much. I made a short introduction. Obviously, you're so well known in the US, I would say, less well known in Europe, but your work has been cited a lot. If you could say with your own words, what were the discoveries, how you treat your patients, how you consider science, by way of introduction, that would be really grateful.
Yeah, thank you, Xavier, for having me on your show. I am Wafik El Deiry. I am a practicing physician scientist who directs the Legaretta Cancer Center at Brown University, where I am also an associate dean at the medical school. I wear a few hats. I have a weekly clinic. My practice falls between treating patients with colorectal cancer, but I also have an interest and specialize in precision oncology. And for the last couple of years, I have been the chair of the International WIN Consortium in Precision Oncology, which is actually an organization that is originated in Paris between the leadership of Gustave Roussy and MD Anderson 16 years ago, and continues to this day to grow and to try and have impact on how patients with advanced cancers are treated using the latest advances in artificial intelligence, in targeted therapies and functional genomics and functional precision medicine. So it's an exciting area. I'll say on the research side, there are a couple of things that I'm well known for. One is having discovered a gene about over 30 years ago now when I was at Johns Hopkins that carries the name WAF1, which has some similarity to my first name. And nobody cared what you called a gene back in the early 90s, but this one turned out to be important. And it gets turned on by a tumor suppressor known as P53 when our cells are stressed or damaged. It has become a part of how we understand how cells stop dividing to repair damage and avoid turning into cancers in our bodies normally. For a number of years, I thought that would be the most important thing I would ever do, which was sort of depressing as a faculty member for years at University of Pennsylvania. But I became interested because I'm a medical oncologist. How do we kill tumor cells? What can we do to kill them, and what can we learn about how the tumor suppressor P53 activation kills tumor cells, especially after treatment with radiation or chemotherapy? And this is how I became interested in the innate immune system because my lab was able to connect the P53 tumor suppressor response and the response after chemotherapy and radiation to something known as a death receptor. Death receptors are induced in tumor cells, and our natural killer cells kill the tumor cells in part through signaling through these death receptors. And as the years went by, I wanted to be able to turn on this host innate immune system. And so we did drug screens to upregulate this pathway, the trail death receptor pathway, with a small molecule in a manner independent of P53, because P53 is commonly mutated in cancer. And so we discovered a drug, a small molecule called trail-inducing compound number 10, about 18 years ago. And that drug has gone by the name of ONC201, dordaviprone, and was recently approved by the US Food and Drug Administration as the first targeted therapy for an aggressive brain tumor, diffuse midline glioma, that carries a mutation in H3K27 gene. There's a mutation there that happens in children and adults with this really aggressive cancer. And the drug made a difference in their survival and was approved. And so, I didn't wake up one day thinking, let me discover a drug to treat brain cancer in humans. But that's what I went through. We knew it had activity against brain cancer, but also that it worked in multiple other tumors. And so we've been asking questions now for at least 10 years, why did it turn out this way? And how can we get this drug and related drugs to work better, not just in brain cancer, but other tumor types? And so I'll say that when the COVID pandemic started, something started to happen in the United States and maybe elsewhere in the world where at universities we were hearing things like research is non-essential. And as a practicing physician scientist, I didn't like hearing that because I felt like, well, there's a global problem. We all have knowledge and expertise, and maybe we can do something to be helpful. And so at the time, Brown University was giving out these small grants to do COVID research. And I looked at that as an opportunity to make a contribution, but also, frankly, to keep my lab open and not shutting down. And because we have expertise in the host innate immune system and cytokines and immune responses, I thought, well, maybe we could learn something about the severity of COVID by measuring things in the blood, and maybe because of our expertise in molecular biology and our expertise with FDA approved drugs, maybe we could learn something about what drugs or classes of drugs out there can suppress infection. And, we discovered a drug class effect with MEK inhibitors that are used to treat cancer. The whole class of MEK inhibitors downregulate ACE2 expression, and this would reduce the ability of the COVID virus to infect human cells. And at the time, we also noticed that remdesivir increases ACE2. And we wanted to do things with that, but by the end of 2020, and we published this initial observation in the summer of 2020. So it was very fast and very early, but by the end of 2020, we had vaccines and the world was now ready to move on, and there was much less interest in some of these other approaches. I should say that as a physician who was taking care of patients at that time, I was very glad when these vaccines came out. I have been accused of being anti-vaxxer and so on and so forth. I do think vaccines have benefits, but they also have risks. And in the case of the mRNA vaccines, this was sort of a new vaccine strategy that was finding its way to the human population. I will say that early on, back in March and April of 2020, when we were planning our experiments on COVID severity and COVID infection, as a cancer biologist and someone who's an expert in cancer and how it develops and how it spreads, and also in treating cancer, I became interested in whether the COVID virus itself has effects on the body's host defenses against cancer. So, it's well known that viruses cause cancer. There are very well established examples. There are other examples that are controversial, but it wasn't very hard for me to come up with the idea of we knew about spike. Could spike or other proteins encoded by SARS-CoV-2 have effects on cellular proteins that are involved in suppressing cancer. There are very good examples with human papillomavirus that suppresses P53 and RB. And, I've studied P53 for over 30 years. We have the tools to interrogate P53 function in human cells, including after exposure to chemotherapy. And so I felt like these are experiments we could do. We could express spike
Yes.
in human cells. We could look at the functional status of P53 and consequences. And we had planned to do these experiments. We got around to doing these experiments in by 2024 actually, and published a paper in I think it was April of 2025 with some results related to spike effects on attenuating P53 activity using different approaches, reporter genes, using endogenous P53 targets, some of which we have discovered, over the last few decades. We looked at sensitivity of cells that overexpress spike protein to chemotherapeutic agents like 5-FU or cisplatin. And we even started to look at the activation of DNA repair in cells, all preliminary stuff, but this is what we found, and so we shared it. When we wrote that paper, uh, as an oncologist, I tend to be optimistic but realistic, and so I'm always looking at trying to see the positive side, trying to see the bright side, trying to ask the question, what can we learn from this experience? And so I found myself in the discussion of that paper that was published last year saying, well, if you want to develop a vaccine against a virus like SARS-CoV-2, and you want to use proteins such as spike, you want to optimize the antiviral effect of the vaccine, but you also want to limit collateral damage and effects on the host immune system, the host innate immune system, the host defenses against cancer, any effects on tumor suppressor genes. These are relatively straightforward things that you can screen for and consider in the design of the vaccine. So that was our contribution at that time. And, if you
Professor,
look me up,
if I
yes.
may, so of course, you have a lot of experience, and your work has been followed extensively and is well, I would say, recognized in the community. But let me ask you one question, because when you look at it from a scientific point of view, there's a lot of individuals who, on social networks or on national media, have been, I would say, I've looked at the increase in cancer over the last 10 years. I mean, it would be interesting to get your point of views of the increase prior to 2020, 20, just at the time of COVID, and since 2020, because we always hear, and I'd love to hear your views on that, here on there could be link between the COVID shots, the mRNA technologies, and cancer. I'd love to get your point of views, whether there are any truth in the increased numbers. The reason why I'm asking this is we have in France conducted a survey on a national representative sample, and 86% of French people are asking the authorities, the regulators, the Ministry of Health, in order to investigate the potential link. And there is some form of refusal from certain scientists to get into this. So I'd love to get your point of view on this. It's an open-ended answer. I don't have any agenda on that.
Yeah, thank you. So, after the publication from last year, sort of linking molecular effects of spike protein and P53, whether the spike came from the virus or the vaccine, um, during the latter part of 2025, I was invited to serve as a member of the COVID immunization working group that is reports to the advisory committee on immunization, the ACIP committee. And so over the summer and the fall, and I really agreed to join that committee to bring my expertise on cancer and to bring sort of my medical experience, clinical research experience, issues with informed consent from the perspective of my field to this, along with the prior research that we have done. So, in the summer and the fall, I started to dig into the literature about COVID and cancer to find out what is out there and to learn about different mechanisms that people have studied. But so, I ended up with my colleague at Tufts University, Dr. Charlotte Cooperwasser, presenting at several meetings, including at an actual ACIP meeting in September. And our presentation focused on some things that are unexpected with COVID mRNA vaccines. We wouldn't be talking, we wouldn't be doing much of what is happening now if, this wasn't part of this, which is the vaccines are rolled out and then there are some unexpected observations or findings that are concerning. So, some of those include the biodistribution of the mRNA vaccines in humans. I mean, I think Dr. Bhattacharya has said for over a year, you don't know what the dose is, and you don't control the biodistribution of these mRNA vaccines in humans. That is absolutely correct. But if you listen to what some say, you'll hear that, well, mRNA is very unstable. It's going to last for a few hours. It'll be cleared from the body right away, soon. But what has been found is that the mRNA vaccines linger in the human body. They've been found in different tissues. They have been found, spike protein has been found in different tissues as well. And evidence of this mRNA vaccine lingering has reached, up to 2 years or more, where it's hanging around somewhere in the human body. So that was unexpected, an unexpected finding. I think, well beyond cancer, there are a number of other uh, side effects or adverse events that have occurred and have affected people, and that are fairly well described, including blood clots, major blood clots, smaller blood clots. Myocarditis is well described. Some of it has been fatal, although rare, but that has happened. I have started to learn about there's a neurotropism of spike. The spike protein of the virus or from the vaccine helps the virus get into the central nervous system, and there are neurological, neuroinflammatory, neuropsychiatric things that have been described. Um, and there are other things. I mean, if you look at the package inserts of the mRNA vaccines, they will tell you that we don't have enough information about fertility. We don't have information about carcinogenicity or cancer effects. And this is up to this date. If you just go to the FDA website and download the package inserts, we started to learn other things that the mRNA vaccines are capable of doing. So, for example, the Nobel Prize winning discovery was the modification with pseudouridine, which makes the mRNA stable. Anybody who's ever worked with mRNA, if you breathe on it, it degrades. And so these pseudouridine modified mRNAs are very stable. But having the pseudouridine modification also predisposes to something known as ribosomal frame shifting, which viruses use because they have a limited genome. They use to get more proteins expressed out of different reading frames in their genome. Well, it turns out that pseudouridine modification increases that effect, which really results in some foreign peptides and proteins that could be expressed that could have inflammatory effects as well. Then another unexpected finding was contamination by DNA and as well as plasmid DNA. The plasmid DNA, we've learned that what was disclosed to regulatory agencies, including the European agency, was missing some SV40 sequences that are
Yeah, that's the
part
work
of
of Kevin McKernan, huh? And
Kevin
Jessica Rose.
McKernan and David Spiker and Phil Buchholz and,
Yes.
and Jess, forgetting her last name right now.
Jessica Rose as well, yeah.
Um, but anyways, so there is, this is unexpected. And this has, the limits were set by the FDA decades ago for 10 nanograms of naked DNA. As it turns out, that's fine, but that doesn't take into account lipid nanoparticles. And as my colleagues point out, the smaller these fragments, that 10 nanograms end up being, billions and trillions of ends that, if they found their way into the nucleus, could insert and have effects on oncogenes or tumor suppressor genes. The plasmid itself and a very large part of the plasmid has been sequenced contiguously. So it's not, people are imagining this, from actual pieces of DNA on a contiguous basis. The majority of the plasmid has been sequenced with depth as well. So, this is concerning. It's unexpected. And as we hear about the different side effects and the persistence of spike, this was sort of the backdrop with looking into cancer. Now, there are a number of mechanisms that could contribute to cancer development, including any sort of insertional mutagenesis things that are going on, or spike effects on tumor suppressors like p53, or immune effects. And one of the things that these COVID vaccines do when given repeatedly as boosters, by about four shots total, you start to have something known as immunoglobulin switching, with production of IgG4, which is immune suppressive. And there is a cohort of patients, I think it was 96 patients with pancreatic cancer, and the ones that had higher levels of IgG4 had poor survival compared to the rest. Now, with cancer, you hear things like there's turbo cancer. You hear a lot of things and see a lot of things on the internet. I will make some points here, which is, so we were interested in what does the world's literature say about COVID infection, COVID vaccines, and cancer. And that's what we started looking into. And so we found numerous case reports and case series and some population studies that address the issue. Now, I am an oncologist who treats patients with advanced cancer. And as I started looking at this, I mean, one of the first cases I saw was a sarcoma in the arm, very close to the injection site.
Yeah.
And, the first time I saw that MRI image, I just asked the question, how does that happen? And as the months have gone by, for that patient, with the clinical description, with the temporal association, there's also this spatial association, geography. It's right there. That to me looks like a smoking gun. Now, there's more. As you look at the different types of case reports, lymphoid and hematopoietic malignancies, there's quite a few lymphomas that have been reported. Maybe, I think, of the sampling of what's out there, it's like 40%, but I don't think that number, that is a very, very preliminary hint. I mean, for me, what's surprising is that it's more than 10 or 20%. The fact that it's 40% suggests some sort of enrichment for this type of tumor. But what seemed to be happening is, again, this geographic spatial aspect of this, with involvement of regional lymph nodes, or some development of head and neck cancer, and some skin cancers that have occurred as well, some breast cancer. In addition, some brain tumors as well have been reported. And so, as I look at this, I come from the oncology world, where, we do clinical trials, testing interventions that are therapeutic or non-therapeutic, and we follow adverse events and report them. And, in the context of informed consent, you don't have to have causality. Causality can take a long time. And frankly, certainly in the setting of clinical trials, a temporal association coupled with some likelihood that there is a relationship raises red flags. And all it takes is one in a clinical trial, one unexpected death. Everybody knows about it. It gets incorporated in informed consent. It could stop a clinical trial, depending on the seriousness of the event, and certainly if there are multiple events. So, at that level, some of what goes on in the world involves epidemiologists and people who like to see large population studies. And of course, that's important, but so are the case reports. Some people look at that and say that's low-level evidence. Not for those people. It's not low-level evidence. It's very, very important. These events have been documented and need to be known. I think up until today around the world, we don't have true informed consent with COVID mRNA vaccines. They don't talk about some of the things. They talk about some things, and I think the package insert will mention, there is a risk of death, which at least it says that, but many other things are not included and could be included. Now, some people out there are pretty paternalistic or worry about how the public interprets things. And I would say that the public is actually very smart and very savvy, and knowing what has happened, regardless of how rare it is important. If something has happened, it's okay to include it in consent, and it's okay for somebody to talk about the risks and benefits with their physician. That's how medicine is done. And certainly here in the United States, our goal is not to prevent anyone from getting their COVID booster. If they and their physician decide that's in their best interest, they should certainly be allowed to do that, and they shouldn't have to pay for it either, if it's in their best interest. But on the other hand, there are things that have been observed, there are gaps in our knowledge, and we need to learn more. Like for me, with regard to the cancer risk and some of the conversations that I've had online with the statisticians and the population level researchers, is well, there isn't an obvious signal. Billions of doses have been given. Well, I would say billions of doses have been given, but not 5, 6, and 7 shots, including boosters, in populations that have been studied carefully. Most of the studies that have looked at outcomes didn't go beyond 42 days. Certainly in the United States, we don't have reliable long-term follow-up that addresses cancer. But then there's something very important, which is really what I believe ultimately, is that there's a range of susceptibilities. There's a great deal of heterogeneity in the population. We're all different from each other, and a risk for cancer is different. And so I believe some people are more susceptible than others. And I think this is where science can help. The risk of having some adverse event depends on the host. Okay. And so that's where science can help. If we know cancer runs in families, we know cancer rates are higher in certain geographies due to environmental exposures or certainly other factors. And so we can be more precise about risk. And again, this is where informed consent could say something as simple as cancers have been reported, they are rare, but we can't exclude that some people are more susceptible. I don't think that's going too far or misleading anyone about anything.
So you want people to be more precise in the way that we report sort of the risk sides and the benefit as well, I suppose. And if I actually read the work and read the publication you've just done very recently, but before we actually get into this, because of the approach you have as a scientist, as an oncologist, as a practitioner, you've been asking questions. According to what I hear, you don't have all the answers. And in essence, you will continue to search. But because you were asking those questions, it looks as if you were attacked, that you were censored because of your, I would say, it's not skepticism. As if you're a real scientist, you know you will have more questions than you will have answers probably in your lifetime. I've been following your work. I've been following the social networks. I've looked at the way you've been treated. And if you could just explain to our audience really how you felt about it when you have such a scientific approach. You don't have an excessive approach. You're not sort of proning for you're really pushing for risk analysis and therefore if you could explain how it came about and how you felt about it.
Yeah, thank you for bringing that up. So the attacks started about a year and a half ago. Our paper about P53 and spike was published, I think, around April, and by June and July of 2020, uh, uh, 2025, the sorry, 2024,
Okay.
the attacks really started to take off on Pr with lots of concerns by anonymous individuals publicly who are concerned about the possibility of fraud and misconduct. And they go back sometimes 30 years. We've been publishing my lab since the mid 90s. And they go after irregularities in loading controls of gels, or sometimes, 20 years ago, there were no rules about splicing in the way splicing bans on gels. Now we have rules, but the rules have changed. They have no attention to significance of any of their concerns. In fact, one of their concerns was about a typographical error in a supplementary table for one of our COVID publications that associated cytokine alterations and macrophage activation syndrome with severe COVID. And, it said incubation when it meant intubation as a measure of severity. And if you read the paper, there is no doubt in anybody's mind this is a typographical error, but they pile it on. Not all of their concerns are actual concerns. Some of them are comments, but I think at first they took it up to like 50 or 60, then they were up to 80. I think they're up to 100, and they're doing this around the clock. They've also attacked the Oncotarget journal before the paper was published, in December and in January, and that led to some censorship. But I will say that it's the public nature of the concerns, which then trigger investigations. So they also write to the government, they write to employers, they write to journals, and everybody who gets these concerns turns around and starts an investigation and is requesting things. And, I can tell you that in a year and a half, nothing's been found as far as fraud or misconduct. There has never been fraud or misconduct in my laboratory in 30 years. And yeah, I mean, there were some mistakes. We will get around to correcting and have corrected some very minor, inconsequential errors. All those citations and knowledge of our work in the field where we're not seeing people publishing papers saying, no, you're wrong, you got this wrong, or whatever. We're talking about papers that have sat there for decades. And so that in and of itself has been challenging. And then there's the attacks online with defamatory comments. I posted some recent example of the words that were used, which, at a minimum, you would say is unprofessional and just inappropriate in a civilized society to degrade and insult and defame individuals
So
online.
why do you think you're the target of such attack? Such attack? Is it because of the way you're doing your work? Is it because you're disturbing an industry, or is it because you're a very recognized and well-known individual in your
Yeah, no, I
field?
don't think it's that. I think, and I am not somebody who has done vaccine research. I'm interested in certain mechanisms and in certain relationships with cancer. And I think the papers we've written have been very balanced in their presentation and in their discussions. We've shared information that we know, and we use what we know to see a future. Where does this go? But clearly, uh, what we have been finding and reporting goes against a certain narrative, and there is interest in suppressing other sides, other narratives that go against what is dogmatic out there now with regard to the safety and efficacy of these vaccines. I mean, big pharma is involved, big pharma profits are involved. PubPeer and Retraction Watch have been questioned in terms of their motivation, their political leanings, their funding. And, I'll tell you, I think part of the narrative that's been gaining momentum since November had to do with this Nature paper that basically came out and said the mRNA vaccines are actually a good thing. If you take them and then take immunotherapy, you will do better and live longer. And I mean, we've seen a lot of papers in Nature. Frankly, I would have liked to see a bit more uh, critical handling of the data and what it means, and a little more rigor. It's written like, hey, this is the best thing since sliced bread.
Yeah, this paper looks more like if it's a marketing paper rather than a scientific paper.
Yeah, and I will tell you. So I've looked at the paper, and what is very clear in the paper is that first of all, they treated patients with melanoma and non-small cell lung cancer. These are the most highly responsive tumors to cancer immunotherapy, besides mismatch repair deficient tumors, like we have in Lynch syndrome, for example. And so that was sort of the starting point. A big question immediately is how will this help the cold tumors that don't respond to immunotherapy? Um, but then if you look at the data in the paper and look at the primary effect of the mRNA vaccines is they trigger an innate immune response that actually causes more PD-L1 expression in the tumors. So in isolation alone, the mRNA vaccines increase tumor PD-L1. Increasing tumor PD-L1 is an immune evasion mechanism that's tumor promoting. That's what it is. Now, it's been argued that, well, yes, but it sensitizes to immunotherapy. So what's wrong with that? I mean, I think, so I've asked, I mean, sometimes these online conversations help you think some things through. So one of the things that I was told was, which I believe, is that both radiation and cisplatin increase PD-L1. But the difference is that those agents, radiation and cisplatin, have anti-tumor effects. And, they've been used to treat cancer long before we knew about PD-L1 or that PD-L1 is increased. And when that was recognized, sure, it's being combined, but, those are anti-tumor agents. The mRNA vaccines themselves are not anti-tumor agents. They increase PD-L1. They use interferon as part of the mechanism. And frankly, interferon is approved in melanoma already. So is it anyway? So I have some issues with that general approach to achieve better outcomes in patients. I think what I'm hearing now is that there's an 800 patient follow-up prospective study that is planned and that will be focused in non-small cell lung cancer. Personally, if you ask me, I'd say, yeah, it's fine to do clinical research. Let's hope the informed consent is accurate and up to date about what we know. And that, the study gets approved by an IRB that has the available information about the effects of the COVID mRNA vaccines on tumor PD-L1, as well as all of the different adverse events that have been reported. And I think in that context, it's up to the way we normally do clinical research, because that's what it is in this case. Um, there is an important point having to do with the risk in the uh, population. I do think there is a variable risk of adverse events in the population. It's already starting to come out in some of the other disease consequences of mRNA vaccines. But I think with regard to cancer, it is we have a gap in knowledge. Where we don't have a gap, so much of a gap in knowledge, is the idea of an effect on a pre-existing tumor. So, the phenomenon of hyperprogression has been reported for several years with cancer immunotherapy. Some tumors appear to grow faster after patients receive cancer immunotherapy. Genes amplifications such as MDM2 and MDM4 that we've been interested in because of their involvement in the P53 pathway, but also EGFR has been implicated in such hyperprogression. So, from the oncology world, it is not such a foreign idea that a cancer could grow faster. In fact, the Nature paper shows the mRNA vaccines increase tumor PD-L1, which on its own is not something that's going to limit the growth and may promote the growth of the cancer.
And Professor, if we go back 1 second to the censoring and to the attacks that you've been subject to, in France right now we are just having a review by the Minister of Health of disinformation and so on and so forth. What step do you think institutions, journals, governments should take to protect people like yourself? To protect your academic freedom, to ensure that there's an open discourse. I can only relate to the restoring gold standard in science that Jay Bhattacharya is actually trying to. First, this is executive order of President Trump.
Yeah, thank you. Thank you for asking that. I do think that individuals like myself and quite a few others who have been targeted, including investigations and retractions, need protection from the attacks by Pr and Retraction Watch. I think there need to be guidelines, there needs to be a discussion from within the profession that includes more than publishers and individuals who are just focused on scientific integrity. I think important stakeholders include, the academic world, who is impacted by these activities. And so there needs to be a discussion and guidelines. Now, with respect to the attacks, I realized a year and a half ago, I wasn't going to immediately go out and hire an attorney. This was bigger than me. And in fact, when I first started to be attacked, I called the director of the Office of Research Integrity at the NIH and, was telling him what was happening. And I learned that about 1,200 scientists at the NIH had been attacked, and some have gone to court, and the First Amendment rights have been upheld. But I think that that still does not make it okay to defame and ruin people's reputations publicly. And, the other thing I learned when I called the NIH was that the NIH has not publicly set guidance in terms of grant reviews regarding, I mean, people who serve on study sections and review grants can always Google people and look at, PubPeer and Retraction Watch and so on. Um, how is that handled? Because it's pretty easy to become very quickly disadvantaged in science funding, which is already very limited and competitive. And similarly, in terms of publishing papers in top tier journals and so on, reputation is important. Like for me, my reputation is more important than any paper I'm ever going to publish. It's more important. And it's not worth it to jeopardize that for any reason. So I do think that we need the government to pay attention. And when I say that I didn't think I would go out and hire a lawyer, I actually think the solution to this problem would include the FBI, the Department of Homeland Security, potentially the CIA, potentially the SEC, HHS, NIH. There's a lot of moving parts to this, and you could say you're crazy. Why do you say that? Some of these people live in other countries, including Germany. It's well documented, and there are a lot of aspects to it between what happens on social media to what happens in the publishing world and in academia. And this isn't something that's going to be solved by any one person. And I think having reform and coming up with guidelines from within the profession is very important. And I don't see that that has happened. I
It's not, it doesn't look as if it's happening. And on top of it, what we're seeing in France is we're seeing those individuals or those individuals that have been targeting people like yourself. They are asking government officials to have protection themselves. So if I take, for example, the Peer mob and so on and so forth, they are seeking their own protection in order to continue conducting let's not call it illegal activity, but let's call it, because let's leave that to the lawyers and to
I mean,
justice.
honestly, the what I have gone through, I have used the word criminal activity publicly online because that's what it feels like.
Yes. And therefore, it feels that, of course, what you're calling for is it's a much wider investigations that would involve the various sort of services of any country and cooperation. Because what you're seeing in France, so what we're seeing in the US, we've had exactly the same thing in Europe with Professor Rao in France, for example, who's been targeted and hassled as well. But thank you very much for sharing with us this element. I have probably one last question for you because I know time's running out. Recently, you've published on social network an opinion with respect to the way forward with this COVID mRNA review. Could you please share with us these elements? Because there's a lot of individuals who were expecting when Robert Kennedy Jr. went into as HHS secretary or Jebet Sharia, that they would immediately pull this mRNA platform. But it's a lot more complicated. And I read your proposal, and I would love it if you could explain this proposal and whether it's got some mileage or whether it's going to be followed, in your opinion.
Yes. So I have said, and I've called it a personal opinion,
Yes.
okay, that there's probably a path to seeing revocation of COVID mRNA vaccine approval by the US FDA for good cause based on emerging evidence of unexpected contaminants from an altered manufacturing process. And I think you know about the process one, process two,
Yes.
with the DNA and plasma contamination being process 2 that was rolled out in the
It's
marketed
different
product.
from the one that they actually got the authorization from.
Correct. And the original one was the one that was used in the clinical trials. Also, the unexpected biodistribution and other biological characteristics in humans that have been noted. I think information on effects of repeated boosters and adverse outcomes, including long term, among different age groups or situations, such as early pregnancy, young men, neuropsychiatric effects, clotting, fertility, prior COVID infection, among others, is really essential to shape guidance and any continued approval. Um, I said that the FDA could warn the various manufacturers of revocation of approval by a certain date in the immediate future if satisfactory evidence of clean manufacturing and safety is not provided immediately or has not already been provided to the FDA. I think whatever evidence is provided should undergo high standards of scrutiny given issues, including technical concerns about the assays used to look at the amounts of DNA and how things like RNA, DNA hybrids, and sensitivity to DNase that's been used could impact the results and underestimate the level of DNA contamination. I think there should be financial and other sanctions towards those responsible. And I think the manufacturers would fall in that category, as would individuals within the FDA or the CDC who may have been involved should appropriate evidence be collected based on the lapses and or inaccuracies in reporting of issues that have surfaced, including many of the things that have been coming out slowly through the work group and the work of ACE and, and many of these things are very relevant and important as far as informed consent.
Sorry, we were just technical glitch probably.
Yeah,
So
yeah, yeah. So anyways, I think, we've basically come up to the end, but the last thing I was saying was uh individuals in society have autonomy and have the ability to say, no, I don't want something to be put in my body based on informed consent. And just for perspective, remember, we're not in the middle of a rip-roaring pandemic. It's 5 years later. It's time we get some answers. It's time we refine the risks in different situations. That's really important. Because I think a lot of the other counter narrative is that safety and efficacy, billions of shots, it's not specific to specific situations or risk categories. And I think it needs to be in the setting of informed consent. This is what physicians and patients have been doing for a very long time. And, physicians take the Hippocratic Oath and they have their patients' interest. And, if they decide a vaccine makes sense, they shouldn't be deprived. But then the other side needs to be addressed. And I think all of this needs to be done with mutual respect and with scientific discourse and scientific progress that is needed. I mean, one of the damaging effects of the censorship and all of the other things we spoke about is it does get into the way of creativity and the scientific process. This isn't how science is supposed to be. We need to let science to be conducted with free thought, free exchange of ideas, and the truth needs to come out,
It gets
whatever
in
it
the
may
middle
be.
of a trust as well. Yes, truth and trust. And
It's truth, but also injustice. I think there does need to be justice,
mhm.
coming back to your earlier question.
And Professor, there maybe my last question, just your point of view on what was the reception that you got from the position, from the opinion you voiced, from regulators? Did you get people who called you and say, Professor, you're right?
I will tell you, I can't remember if I mentioned this, but I heard from a representative in the Japanese government,
Yes.
um,
He,
by the name of his Kazuhiro. I'm
yes,
forgetting the
thank you
last
for
name.
your work. Yes.
Yeah, no, but I mean, he himself had a diffuse large B-cell lymphoma 3 years
Yes.
ago. It metastasized to his tonsils. It was biopsied. Spike was found in it and not nucleocapsid protein. So it's from the vaccine. Tumors should not have spike protein. And I think we do have major gaps in tissue analysis and analysis of autopsies to get at some of what's been going on. And we need more of that.
But you didn't
Um,
hear anything from the FDA or from the CDC or from the NIH?
I can't say too much because I'm on this advisory group, which is continuing to work, and everybody's seen the
The people.
paper, everybody's aware of just everything we spoke about. So I expect more things to happen as a result of this and the other work that is being done. So thank you very much. I need to go
And thank you
to an
very much,
important.
Professor El Deiry, for your time and for this very long interview. Have a really, really nice day.
All right, let me know when it comes out.
Yes. Thank you.